Welcome to all. I am R. Mahalakshmi from Central University of Tamil Nadu. Today we are going to discuss about role of microorganism in bioremediation. This is part B video. Part A video has already been presented by Navyata. On that video she mentioned about what is bioremediation, different types of bioremediation including biostimulation, bioattenuation, bioventing and all. So today we are going to look a little bit deep onto the topic. So here in the picture itself, it shows like microorganism, how microorganism clears the oil spilling or like oil decontamination in a contaminated area. So here it like just shows it engulfs the oil, like oil or the other organic matter. Then it will digest using its mechanism, converting it into harmless carbon dioxide and water. Then it releases the harmless compounds like carbon dioxide and water. So in this picture, we can see that it conveys a basic message that there are different pollutants in our environment, either man-made or from natural sources. These pollutants will transform to other pollutants by either chemically or by reaction to sunlight. As we all know that there are primary pollutants, then primary pollutants, they'll transformed to secondary pollutants. From that, secondary pollutants are produced. So this will have a huge, serious impact on weather, on humans, and also to birds and animals, etc. So how this happens is like if there is an emission from industrial waste or from a like wastewater treatment, that will lead to the formation of these pollutants, harmful pollutants. Then these pollutants will disperse into our environment, atmosphere and everything. Then they will like gradually they will increase their concentration. There will be increase in their concentration. So in order to clean up our environment, there are different methods to reduce these pollutants in our environment. But bioremediation is one of the method which is sustainable and eco-compatible. So here we are seeing that bioremediation of marine sediments contaminated by petroleum hydrocarbons. So as we all know that petroleum hydrocarbons are one of the most widespread and heterogeneous organic contaminants affecting marine ecosystem. The contamination of marine sediments or coastal areas by these kind of PHCs represents a major threat for the ecosystem and human health. So it calls for an urgent and effective need to like find a remediation solution. So like as we all know, petroleum hydrocarbons contaminating aquatic environments are mainly composed of three classes of compounds, which are alkanes, olefins, and aromatics. So apart from this, there are other chemical contaminants such as heavy metals, metalloids, aliphatic, and then halogenated compounds, which are introduced into our marine environment by multiple sources. So this will cause a negative impact on our marine environment by like high anthropic pressures and reduced hydrodynamic regime, which may lead to the high accumulation of chemical contaminants in the sediment. Then another issue is that like half of our population depend upon seafood. So when they consume this seafood, like fishes and everything, it will create like these pollutants will accumulate on our biological tissues and it will create another intoxication problem and everything. So like different physicochemical techniques have been developed to reduce this pollutant concentration from water. However, there is an urgent need to find sustainable and eco-compatible solution for the remediation of these contaminant sediments in situ. There comes our bioremediation. It is an environment friendly and low cost strategy which relies on the ability of microorganisms including prokaryotes, fungi and microalgae to reduce contaminant concentration and their toxicity. So there is one more thing regarding these PHCs. PHCs like generally they have poor solubility on water. So by this thing they'll show a long-term persistence on the bottom of the sediments with a significant negative impact on benthic aquatic communities. So general scheme of bioremediation strategies involving different microbial taxa. So these are different microbial taxa, bacteria, fungi, microalgae. Also plants can also be used for the bioremediation, but our topic is to discuss about the microorganisms which are involved in bioremediation. So natural attenuation, it is like bioremediation process only, but bacteria, like slowly with the time they will degrade or they will remove the pollutants. Biostimulation is that we will add, like commonly microbial based bioremediation strategies are there. It will include the acidic, addition of the specific compounds to stimulate the microbial assemblage, that is called biostimulation. So in addition to that, it can also display useful biodegradation or detoxification capacity also. So nutrient will, like when we add some kind of compounds to this, it is called biostimulation, a kind of bioremediation. Also microbial addition, it is called bioaugmentation. So particularly for the bioremediation, the use of autochthonous microorganism is expected to be more effective and ecologically friendly. So what is autochthonous means? The microorganisms that are inhabitant of the same environment, that are called autochthonous. Like they are better adapted to this specific local environmental conditions than the allochthonous microbes, which may require the manipulation of the natural environment to maximize their performance. So as I already mentioned, it will include the biostimulation, bioaugmentation and other methods. So that is changing the oxygen conditions or nutrient concentration, etc. So among the bacteria involved in the bioremediation processes, most belong to the genera Alkaligens, Acromobacter, Acetinobacter, Alteromonas, Arthrobacter, Burkholderia, Bacillus, Enterobacter, Flavobacterium, Pseudomonas. Genera such as Alcanivorax, Marinobacter, Thalassolitus, Cyclocalsticus, Olicipira include hydrocarbonoclastic bacteria and are known for their specific ability to degrade hydrocarbons. As I already mentioned, polyhydrocarbons are the major contaminant in marine environment. So next is microalgae or cyanobacteria. We can also use microalgae for bioremediation. So these are the classes that have been used for classes of microalgae that have been used for bioremediations. Spirulina, Chlorella, Spirogera, Cynodesmus, Oscillatoria, Chlorococcum, Senecocytis, Nanochloropsis, then Selenastrum. So among these microalgae, mainly green algae belonging to the genera Selenastrum, Cynodesmus or Chlorella, they have been demonstrated to be effective in the degradation of polycyclic aromatic carbohydrates such as naphthalene, phenanthrene and pyrene, and the immobilization of heavy metals. So heavy metals like chromium, mercury, they have shown like they can reduce the bioavailability of these pollutants in the marine system environment. So here is a picture that shows the mechanism of bioabsorption of heavy metals and the detoxification by microalgae. So these heavy metals in the surroundings can enter into the cell by various methods. One is cation exchange, another one is complex formation and chelation. So another method is they have these microalgae have some exopolysaccharides compounds in the cell surface that can mediate the uptake of the contaminants on the cell surface and into the cell. So thus by enabling these microalgae to remove the toxic compounds, thus by they'll reduce the bioavailability. So here by covalent bond it will attach to the cell, and by ionic bond also it can attach to the cell. So once they entered into the cell, there is two things that can happen to the heavy metals. One is either they'll transform, biotransformation will happen by redox method or by methylation. These inorganic heavy metals will turn into organic heavy metals, or they will be internalized, like this internal like in the vacuole these heavy metals got sequestration and compartmentalization will happen and they will be immobilized. These heavy metals got immobilized in the cell itself. So thereby they can reduce the bioavailability of these heavy metals in the surroundings. So these adherent or internalized molecules belonging to, like, they have been classified into a class called phytochelatin class. So now we are going to see the factors that affects the bioremediation of sediments polluted by the petroleum hydrocarbons. So we know that the pollution of marine sediments by petroleum hydrocarbons is a widespread concern that affects the coastal regions and of the globe, and it represents a major concern for the uh, like ecosystem biodiversity, its functioning and everything, like overall health of the ecosystem. So when compared to the in situ and like laboratory studies, biodegradation efficiency of petroleum contamination sediments can be like they can be increased by enhancing the biomass or the activity of hydrocarbon degrading microorganism through biostimulation, bioaugmentation strategies. But before that, understanding the key factors that influence the microbial growth, their metabolism, and the hydrocarbon degradation is crucial to design an optimal bioremediation strategy. So for that, like the sediment grain size, like our marine sediment grain size is important. It can affect the bioremediation yield by influencing the availability of hydrocarbon from the microbial attack and diffusion of the nutrients and dissolved gases that needs to support the microbial metabolism. Also, nutrients for these microbes to perform good is very important. So it seems that nitrogen and phosphorus is a limiting factor, or they are the limiting nutrients for this uh, for this bioremediation. Then the pH of the environment influencing by microbial metabolism can play a role in affecting the bioremediation performance, which typically is effective at pH value around 6 to 8. Then the oxygen concentration. Oxygen concentration is another factor influencing the bioremediation process, as most of the petroleum degrading microbes identified so far as aerobic. So there are two types of bioremediation also. In aerobic and anaerobic condition, bioremediation can happen, but most of the bioremediation petroleum degrading microbes are aerobic. So uh, along with this, the like if an oil spill happens, the surface area can also affect. So compared to one large oil slick, like for microbial degradation, it will give more surface area to numerous small oil spills. Like you understand that the surface area can also influence the bioremediation, the speeding up of bioremediation. So in this picture, we can see the like water column and also the stratification of the sediment layers. So here it shows the redox potential positive to negative, it comes down. Then the oxygen concentration from aerobic, it will come down to anaerobic, that is anoxic. In between, it has a suboxic, like the bacteria that can tolerate the low oxygen level can grow in this region. So like with the in the presence of oxygen, aerobic degradation can happen. Then denitrification, ion reduction, manganese reduction in the suboxic level. In anoxic level, sulfate reduction, methanogenesis can happen. So in the next slide, we are seeing types of bioremediation. Types of bioremediation can be classified into, like broadly, they can be classified into two types: in situ bioremediation and ex situ bioremediation. In situ bioremediation is nothing but on spot, wherever the contamination happened in the environment itself, we are cleaning the or reducing the contaminants. So they can be again classified into engineered bioremediation and intrinsic bioremediation. So again, they are classified into biosparging, biostimulation, bioaugmentation, bioventing, natural attenuation, and bioslurping. In this, biosparging is nothing but giving aeration or like oxygen molecules to increase the microbial activity. All these techniques are used to improve the bioremediation method. So in ex situ bioremediation, it is happening like we are collecting those sediments or those solid surface soils to another place. In that another place, we are creating an environment that can possibly give the maximum result of bioremediation. So two types of bioremediations are there: solid phase bioremediation and slurry phase bioremediation. In solid phase, there is biopiling, composting, land farming, biofilter, and again in slurry phase bioremediation, there is bioreactor. In bioreactor, we can see most of the wastewater sewage treatments, we can do it in bioreactor. So in this next slide, we are going to see the mechanism of bioremediation. That is, two types are there, aerobic and anaerobic. Here, polyaromatic hydrocarbons are degraded through oxygenase mediated activities. So there are two enzymes. Here I have included a picture that shows the mechanism of bioremediation in aerobic condition. So two enzymes, deoxygenase enzyme and cytochrome P450, is two like classes of enzyme that has a critical role in the bioremediation, a process that utilizes the biological organism to degrade the pollutants. Then like they will convert these hazardous petroleum hydrocarbon contaminants to non-toxic simpler components such as carbon dioxide and water through oxidation process under aerobic condition. So it occurs via a sequential oxidation process. So first one is like they are particularly effective in degrading the aromatic hydrocarbons. These efflux pumps will help in the uptake of these hydrocarbon, petroleum hydrocarbon into the microbial cell. Then from there, they'll undergo the oxidation process through deoxygenase enzyme and then cytochrome P450 enzyme, P450 oxidases enzyme. They will add oxygen to these compounds. So this deoxygenase enzyme can break them down into less toxic intermediates that can be further metabolized by other organisms, like other metabolic pathways. Here we that is connected to cytosolic fatty acid metabolism. So after that it will be going to TCA cycle. Then it will release as this petroleum hydrocarbon is released as non-toxic compounds such as water and then carbon dioxide. So and also here two genes like encoding alkaline monoxygenase and NDO encoding naphthalene dioxygenase, they have been identified as some of the genes that genes get activated when this petroleum hydrocarbon is introduced into the cell. So these are some of the examples for aerobic bacteria that could perform bioremediation under aerobic condition: Bacillus, Pseudomonas, Pingomonas, Flavobacterium, Nocardia, Rhodococcus, Mycobacterium, etc. that can degrade a variety of complex organic compounds. Along with this, pesticides are also needed to be reduced. It also comes under bioremediation. Alkane hydrocarbons and polyaromatic compounds have been shown to be degraded by this enzyme. So again, Nitrosomonas europaea is capable of degrading benzene and a variety of halogenated organic compounds, including trichloroethylene and vinyl chloride. So here is a picture that shows the action of Eudionella sequensis. Eudionella sequensis is a bacteria that is currently used for the recycling of PET plastics. PET plastics are polyethylene terephthalate plastics. These bacteria can use the PETase, that is AAS means enzyme, right? Polyethylene terephthalates enzyme, and also MHETase. MHETase is nothing but mono 2 hydroxyethyl terephthalate enzyme that that can break down this plastic into terephthalic acid and ethylene glycol, two compounds which are less toxic. So this is the mechanism through which, like, these are some of the examples through which they can do bioremediation in aerobic conditions. So now coming to anaerobic degradation of pollutants. So as the name states, anaerobic microbes will degrade the pollutants in the anaerobic condition. Then these are some examples for the anaerobic microorganism and the like pollutants they degrades. Dichloromonas aromatica has the ability to degrade benzene anaerobically and to oxidize toluene and xylene. Phanerochaete chrysosporium. It is an fungus, anaerobic fungus that exhibits strong potential for bioremediation of pesticides, polyaromatic hydrocarbon dye, trinitrotoluene, cyanides, carbon tetrachloride, etc. Then microorganism dihalococcate species are responsible for the anaerobic CO2 and H2O bioremediation for toxic trichloroethane to non-toxic ethane. Then Pestaloteopsis microspore is a species of endophytic fungus capable of breaking down and digesting polyurethane. So this makes the fungus a potential candidate for bioremediation projects involving large quantities of plastics. So usually in anaerobic conditions, microorganism will ultimately metabolize organic contaminants to methane. So only a limited amount of carbon dioxide or a trace amount of hydrogen gas is produced. So in the anaerobic condition, instead of oxygen, they will use nitrate or sulfur or other elements as their electron acceptors. Then hydrogen is typically used in this reaction is supplied indirectly through the fermentation of organic substrates. Then I have given a figure like that during the anaerobic biodegradation of chlorinated compounds, it shows the sequential removal of chloride ion. PC is nothing but perchloroethylene, and this is perchloroethylene. When one chloride is removed, it is converted to trichloroethylene. Then cis DCE dichloroethylene. Then it is converting to vinyl chloride. Then it is converting to ethene. Like this, a potentially harmful contaminant is sequentially it is converting to or transformed to like a compound which is not harmful. So a final degradation product is ethene. This is one example for the anaerobic degradation of pollutants. So one of the ex situ technique is bioslurry. So as we all know that bioslurry involves the treatment of a contaminated soil in a controlled bioreactor. We know what is like bioreactor or bio vessels, such as like we can do like batch culture is there, fed batch culture is there, continuous fed batch culture is there. Like this bioreactors are there. With the help of this bioreactor, we can reduce the we can grow the microbes in that, and then we can reduce the contaminants in them. So in a bioslurry treatment system, these nutrients are like there are several nutrients. The nutrients can be added into the bioreactor at a definite time in definite interval to enhance these microbial activities to degrade the hazardous contaminants. So these bioslurry reactors designed with the various processes, that is we can control and we can monitor temperature regulation. Then we can we can take the sample in and out to see whether like how much the concentration of contaminants and the concentration of metabolites and everything. So this all means it is ensuring a maximum efficiency in bioremediation. So it is a ex situ technology that can be used for the bioremediation of problematic sites when the less expensive natural attenuation or stimulated in situ bioremediation are not feasible. So in this bioslurry input, we can input the polluted soil, water, aeration. A good aeration system should be there, microbial consortia, Consortium means like a group of microorganisms that can help in the bioremediation. Then nutrients, biosurfactants, etc. Then operational parameters. We can fix the operational parameters. Then these are the mechanism of degradation. How these bioorganisms, like how these microorganisms are biodegrading these contaminants. So absorption to desorption in liquid phase, biosorption happens, and like oxidation of heavy metals to like inorganic to organic compounds and everything. So as an output, we will get decontaminated soil, wastewater, biomass, CO2 byproducts. So this is a like it results like it obtained when microbial consortia of Bacillus cereus and Pseudomonas putida were used. So the efficiency was in between 79.35 to 99.73% in a slurry bioreactor after 49 days. So next is genetic engineering for producing a strain or microorganism which has maximum efficiency in bioremediation. So microorganism which can degrade different kinds of hydrocarbons that have salt tolerance, biosurfactant producers, they can be genetically modified by using genetic engineering technique to achieve a maximum efficiency in bioremediation. So there are several genetic ways that have been developed and used to optimize the enzymes, metabolic pathways or organism with respect to bioremediation. The essential strategy of using genetically engineered microbes in bioremediation is to design such novel strains which have the ability of degrading high molecular weight polyaromatic compounds. So here I have included a picture that is an example for the genetically modified Pseudomonas putida KT2442. So in this they have taken 4 different strains like strain A, strain B, strain C, strain D, which is capable of degrading each, like one is capable of degrading different compounds like camphor, octane and xylol, naphthalene. Here what they have done is they have isolated the gene which is responsible for the degradation, and then they have gone for recombinant DNA technology. They have created a strain G which is capable of degrading camphor, octane, xylol and naphthalene at once. Then like they have did this in Pseudomonas putida KT244, and then Pseudomonas fluorescens HK44 represents the first genetically engineered microorganism approved for field testing in United States for bioremediation purposes. It is an example for the first genetically engineered microorganism. Then like it also has, this strain also has an introduced Lux genes as a reporter gene fused with an naphthalene degradative pathway, thereby allowing that to show bioluminescence as it degrades specific polyaromatic hydrocarbons such as naphthalene. So so that we can observe the bioremediation process by the detection of light. Then like for the creation of superbug, oil-eating superbug, Indian-born American microbiologist, he was awarded US patent, Mr. Anand Mohan Chakrabarty. Thank you all for listening to my presentation. Thank you, Kavya ma'am.